Good morning, everyone. My name's Chris Ward. I'm head of precision and translational medicine for oncology and cell therapy at Takeda Pharmaceuticals, and I'm based in Cambridge, Mass.
Great. Eric?
Hello, all. I'm a medical oncologist, and I oversee early clinical development and translational oncology for Merck.
Terrific. Sam. Hi, Sam.
Hi there. I'm
Good morning, everyone. Oh, sorry.
Sorry, guys. Kulkarni.
Good morning, everyone. Sam Kulkarni, or for this panel, I'll go Samarth so that we avoid confusion. It's a pleasure to be here. I'm the CEO of CRISPR Therapeutics.
Terrific. And the other Sam.
Sorry. And I'm Sam Waksal. I'm an immunologist, but I am the CEO and chairman of a new biotech company that does a lot of cancer work, amongst other things, Graviton Biopharmaceuticals.
Terrific. And of course, last but not least, Todd.
Todd Golub. I'm a pediatric oncologist by training, and I'm director of the Broad Institute of MIT and Harvard.
In terms of cancer, there's still so many cancers that are left untreated, but where are we today in terms of state of the art, state of technology, state of the disease?
That wins the award for biggest question in oncology ever. I think, Andy. My own view, and I'd be interested in others' perspectives on this too, is that it's clear that we're on the right path to a solution for eventually all patients, to the extent that, I think the concept of precision cancer medicine, which used to be kind of theoretical, is no longer theoretical. I think it's clear that the idea of using the molecular characterization of a patient's tumor to guide therapy is going to be effective, and eventually will not be optional. It will be something that all patients benefit from. And the idea that the immune system can be a powerful tool against cancer is no longer theoretical, because we have obvious evidence of efficacy in some patients. So I think we're on the right track, and one can see a path to continuing to make progress there. But the reality is that most patients still don't benefit from the precision cancer medicine rubric. I think personally the answer there isn't that, we need a different rubric. It's rather to stick with the concept, not lose sight of, for example, the clearly driving oncogenes that drive cancer but are classically considered undruggable. I think we can't take that as an excuse to not target them, because there will be efficacy there if we can figure out how to develop effective drugs or gene therapies against them, and continue on the current path. I would say we're 25% of the way there.
Eric, maybe, I just taking off from where Todd left, I've been struck by the ends of the spectrum, and maybe this is not how you see it. In one end, you have these targeted oncogenes, and the other end, you have this completely non-targeted immunotherapies. Is there a reconciliation between the two?
Well, I think it may be a little bit of semantics. The immunotherapies, of course, have targets, right? They happen to be immune-based targets, but they're, some of the most effective ones, like Keytruda, are monoclonal antibodies, of course, which are highly specific. But I think, I'll echo what Todd said. I think take KRAS, right? That a few years back, we knew that was a cancer driver, yet we really struggled to find a drug. And now we have an approved drug with lots of investigation there that I think, that will, right now we have a G12C drug, right? That there's a lot of other patients with other mutations. And I suspect over the next few years, we'll have drugs that cover those as well. So I think we're making steady progress. And I agree with Todd, we shouldn't give up the paradigm of trying to understand each individual patient, what's the most effective treatment for that patient.
Andy, can you hear me now?
Yeah, terrific. Alise,
Yes.
Go ahead.
Great. Sorry about that. The audio on my computer stopped working at 7:00 this morning. So a little bit of technical problems. Thank you for doing a terrific job standing in. So we've got a lot to talk about, not a lot of time left to talk about it. Let me start. And so has everybody introduced themselves? Okay,
Yep.
Great. Sorry about that I missed the beginning part. So let's start with the first question. The first tissue agnostic biomarker based approval for a drug in oncology is something Eric and I know well. It was the approval of Keytruda in patients with metastatic cancer whose tumor had evidence of microsatellite instability or mismatch repair deficiency. And the initiation of that whole idea was actually by Luis Diaz, who was then at Johns Hopkins and is now at Sloan Kettering. And it was the first investigator grant that we approved for Keytruda. Now, one of the most talked about trials out of ASCO was the early data showing a remarkable 100% complete response in about 15 patients with early stage rectal cancer with mismatch repair insufficiency who were treated with a PD-1 in lieu of chemo. And none of them had to go on to date to radiation and surgery. Now, I'll combine that alongside with a current push by Dr. Pazdur to encourage companies to change the paradigm for accelerated approval in late-line cancer to going into randomized studies in first-line or earlier cancer. So what are the key takeaways from the results of this small but remarkable study? And do you think we're going to see a paradigm shift that Pazdur is pushing for?
I don't think we're going to see a dramatic paradigm shift. And more importantly, I sort of worry, I love the fact that we're absolutely right. Precision medicine isn't going away, and we're doing the right things moving forward, with the caveat that when we look at a 500 patient study, I worry now that if 50 patients really respond and the rest don't, does that mean the drug didn't work? No, it means that we're getting a probability effect in cancer, that each and every patient is like an inbred strain of mice, and we're getting different responses in different patients. And we finally have to try to understand how some of our drugs work. And missense repair deficiency was a great approach. I think if we increase those, I'm not sure we'll see 100% response rates. And I'm not sure what happens when we have metastatic disease, but we're learning more and more about what some of our drugs are going to do. And it's going to change, that's going to change the paradigm. I think it's going to be a probability effect sometimes, only because we don't understand in total what's going on in these diseases.
Todd or Eric, comments about what you think we can learn about immunotherapy and early disease?
I'm happy to weigh in on this one. I mean, if we take at face value that this small trial really had a dramatically different response rate than in previously treated patients, let's just take that on face value. To me, the question is, okay, we need to understand why is that? So yes, the approach, and it probably will happen, to say, okay, let's do a lot of clinical trials in previously untreated patients, and some of that presumably should occur. But I would hope that that will be accompanied by really a deep characterization of what is it about the immune system, like the T cells that are infiltrating tumors before treatment and after treatment. If we really understood that, that would give a clue as to why is the response rate higher in patients. So I hope we don't skip that kind of fundamental step. But Eric.
Yeah, thanks, Alise. So it was a very interesting study, of course, and I think maybe I have sort of three high-level comments about it. One is it was a pretty gutsy study. These are patients who can be cured by conventional radiation, chemotherapy, surgery, albeit with some morbidity there, right? So but you're taking a patient, they're consenting to a treatment that you don't know, right? I mean, there's, of course, there was the data that you mentioned that in the metastatic setting, anti-PD-1s are quite effective, but still, you're taking someone who has where you've got a some chance of cure, and now you're not so sure. And obviously it turned out very well. I think the two other points I would make is that this notion of moving immunotherapy up to earlier lines of treatment now has been going on for several years. And there are more and more examples of where moving to a neoadjuvant or adjuvant setting, with an immunotherapy, even a monotherapy immunotherapy can be quite effective. And so I think part of this may be that a patient's immune system is just in better shape when their disease is an earlier stage. And so I think that I do think we'll see more trials like this, where there's an intent to move to earlier lines of treatment and treat patients who could be cured by other modalities, but where those other modalities have morbid effects that you'd like to get rid of. And then the last thing I'll just point out is that I think an important part of this, of course, is, that this is a really nice predictive biomarker, a very effective predictive biomarker. And I think, there are other settings where one can envision similar types of things. One of the ones that I think is interesting is Hodgkin's disease, where early on, I think it was Margaret Shipp, probably deserves a lot of credit here.
Right.
She was interested in looking at anti-PD-1s in that disease because it was recognized that there are chromosomal alterations at 9p24 that are very common in Hodgkin's patients, and those result in upregulation of the ligands for anti-PD-1s. The ligands are PD-L1 and PD-L2. And indeed, when these patients are treated with anti-PD-1s, their response rates are very high, 90%-ish. So in that disease also, I think we're moving towards seeing if we could actually minimize or perhaps someday even replace first-line chemotherapy in that setting, again, with the hopes of it being a more tolerable treatment.
So we're going to come back to immunotherapy again, because I think there's more to talk about there. But I'm going to take a detour over to cellular therapy, because I think we've got a couple of panelists who spend a lot of their time thinking about it. So there's been a lot of data over the last year switching from autologous to what we'll call off-the-shelf therapy in hematologic malignancies, including allo-T, NK, gamma delta T cells. Certainly, we're seeing responses. I think the jury's still out on durability of effect. What do you see as the most promising approaches? And that's Samarth or Christine?
I'm happy to jump in, but Christine, please go ahead. I'll add.
So I was really excited at ASCO this year about the first clinical data with gamma delta T cells with Adicet. And I think the early data is very, very encouraging. What's really exciting about these cells is that it's the ability to go after tumors that may be able to downregulate the immune system. So class 1 MHC, because these cell types are able to respond to tumors in the absence of that. So very exciting days. And I think it's so early. There's a lot of different ways this is being targeted. At Takeda, we recently acquired 2 companies in this space. So the Gamma Delta Therapeutics and Adaptate, which has more of an engager approach. And I think it's an exciting way to target in an allo way to help us bring these treatments to patients in a way that's more convenient.
I have to agree with Christine. There's a lot of exciting things happening in the cellular therapy space. We may be in some ways at the trough of disappointment. If you think about the early excitement and then some early validation and some disappointment, but this is all going to come back in a big way because what we're seeing is very exciting data. I think 3 or 4 years ago, if you ask someone, are you going to see responses in solid tumors with CAR T, autologous or allogeneic, people would have said, no way. And this week at CRISPR, we just reported that we had a complete response in renal cell carcinoma, metastatic renal cell carcinoma with an allogeneic CAR T, which has gone on 18 months. And I think, in the moment, a lot of these things are lost, but the significance of it is huge as we move from, the paradigm of only small molecules or antibodies to cellular therapies. And it's hard to say which ones may be most effective between the CAR Ts, NK cells, or or say gamma delta T cells or TCRs. But I think what we're also finding is all of these can be engineered in many different ways. We're entering an era of continuous engineering of these cells, and not just the external markers like PD-1 or external receptors like TGF-beta, but internal edits that make these cells much more powerful. So we're just in the early stages here, but I'm quite bullish about cell therapies, particularly allogeneic cell therapies in cancer.
So the patient that you just talked about, what was different with the CAR T that was given to that patient versus many of the CAR T programs that have failed? Do you have a hypothesis? Was there something that you engineered into the T cell, something different about the patient?
Yeah, I'll come back to what Sam was saying early on is, I think what we found in this patient was they weren't heavily pretreated. They didn't have 5-6 lines of therapy. They had 3, but also the tumor was more immunologic, and I don't know how to exactly characterize and define it, but it's the CAR Ts trafficked in there and they were able to get to the tumor. And I don't know if it's probability, like Sam said earlier, or if it's something that we don't understand fully about these patients at this point, but certainly we're trying to do more work there. And we're seeing that across the board, not just in our trials, across allogeneic trials. There seem to be some tumors or some patients that respond much better than others, and that is different from autologous therapies, where more so the success is dependent on whether the patient themselves have a strong immune system to begin with.
So before we go back to immunotherapy, and since we're on cellular therapy and we know, the resources that it takes for cellular therapy, certainly autologous, but even allogeneic, how are we going to make these advanced therapies affordable, even in countries like India? And I'll point out that this week there was a report about a company called Immunel, which is dedicated to bringing cellular therapies to India. So I don't know if you want to, I know this is something that's near and dear to your heart, Samarth, if you want to comment on how you think we're going to be able to make this available around the world in the future.
Yeah, happy to start there, and obviously many others on this panel are experts in this, but hugely passionate about bringing these CAR Ts to countries globally, especially countries like India. And I just actually had a discussion with Immunel, which is quite remarkable. They're trying to do decentralized autologous CAR T manufacturing at a much lower cost than what we see here in the US and in Europe. It's a tenth of the cost. But I think the other approach we're taking is to take allogeneic cell therapies and make edits to them so that the expansion of these cells is 100-fold what we see with the first generation. So our cost of goods, are already in the $20,000-$30,000 range, can go down to maybe $5,000 a patient dose. In which case, you can make this a reality in China and India and many parts of the world. I think it's hard to enter some of these markets. That's why we're having discussions with companies like Immunel or others that are emerging. But I'm very hopeful that with allogeneic or with decentralized autologous therapies, you could get to a very low cost of goods.
I think we have a question from the audience. Olga, are you available?
Yes, thank you. Thank you, Alise. So my question actually is for both immunotherapy and cell therapy. So given largely underwhelming results from next generation IO drugs, should we put more resources into targeting tumor cell stroma interactions that are leading to exclusion of T cells, or creating unfavorable tumor microenvironment in a way to enhance immunotherapy?
That's a great question. And it's a great question because tumors play different games, and pancreatic, ovarian tumors create sometimes a fibrotic shield that keeps a T cell from getting in the way it might in another tumor. And we know whether it's cellular therapy or immunological approaches to cancer therapy, the tumors respond differently, and we are learning why. So I think we are going to see in the future, and it's really going to add on the question of how we get this to the world. We're going to have to add on small molecules, perhaps, that degrade the fibrotic shield in some of these cancers, that allow the cellular therapy or the immunological therapeutic approaches to work. And some tumors will respond better than others. And it's our job right now to learn why there will be responsivity in some, what the markers are that will teach us that, and what we have to do in others.
The other thing I would add to this is we know that when these drugs get into the tumor microenvironment, it's very immunosuppressive. So even though you can get past the fibrotic shield, I think switching the balance of the TME, the tumor microenvironment, to be more pro-tumor killing. And I think there's a lot of really interesting mechanisms targeting the macrophage space. I think about CCR8 and other ones that I think can play a role here as well. And I think it's up to us to really think creatively about these combinations early in the development cycle for these drugs.
Yeah.
Thank you. So, that takes us back to IO. And it's really been remarkable. It's been over a decade now since the CTLA-4s and PD-1s were brought into the clinic. And I think there was such re-excitement about immunotherapy after so many decades of failure. And everyone thought it was going to get easy. Well, it hasn't been easy. And there really hasn't been another breakthrough drug. There's LAG-3. TIGIT is still, I think, an open question. So how are we going to make it? Clearly there's more work to be done in IO. And is the issue that we haven't had biomarkers? Because I think a lot of what made us successful with PD-1, not in all tumors, but in some, was having some of those biomarkers, either for mismatch repair, as we talked about, or even just PD-L1 expression. Or are there better ways to make the translational and preclinical models more predictive? Because the other thing that's been clear is they are not predictive. Anybody want to take that?
I'm happy to weigh in on the preclinical models question. So one interpretation would be, well, mice aren't people and they're bad models kind of categorically. I'm more of the opinion that, I think as Eric alluded to also, immunotherapy is no less a precision medicine exercise than small molecule oncoprotein targeted therapy. And that the fact that the field relies on a small handful of transplantable syngeneic mouse tumor models to drive preclinical evaluation is problematic, because those models don't represent the breadth of molecular diversity that we see in human cancer. So I could be wrong, but my own view is that this is not a mouse versus human problem per se. It's the fact that this very small number of mouse models, syngeneic mouse models of cancer that we have, are good representations of that flavor of cancer, but generalizing that to other tumor types that are in some cases radically different molecularly is problematic, in the same way that we wouldn't think about using an inappropriate xenograft model, let's say, for a targeted therapy to an oncoprotein if we didn't have the right model for that particular strategy. I think that the same holds true for immunotherapy.
Eric, do you have comments on? That, and what you're saying leads right back into the biomarker piece, because then it says you're going to need some sort of biomarker to predict what those tumors are in the clinic that are likely to respond. Eric, do you have anything to add? And I'm wondering, I know Merck has some macrophage, some targets that are directed, in fact, towards the macrophages that were mentioned before. I don't know if you're using a biomarker to try and predict where that might work.
Yeah, we are. So, but the answer to that is yes and yes. So you're right. We're a nice diverse internal pipeline that includes both efforts to extend the efficacy of Keytruda, as well as pursue targets that, that where you would have monotherapy activity with the agent on its own. And in every case, we're looking for potential predictive biomarkers. I can mention, so you mentioned TIGIT and LAG3. We have both of those compounds, our own versions of them. And, we've looked hard to try, you might think, okay, expression of LAG3 would be predictive of the combination. It really adds nothing beyond expression of PD-L1 when we look. So it's not, that's always that simple in the combinations to sort of identify a predictive biomarker that, that you think would work, perhaps beyond the ones that you already have. The other thing I'll comment that one of the fun things about having Keytruda is that lots of folks want to do combination studies. So we have a group that we call our external collaborations group that I think has probably close to 300 studies right now that involve very diverse mechanisms. One of my favorites, and it's public, is there's a group that makes devices that project an electric field. They actually have an approval in glioblastoma,
Right.
and the hypothesis is that this the mitotic spindle is polarized, and that cancer cells are more susceptible to magnetic fields. So we have a combination study with them that I think, because they think this may also be immunogenic. The data is still pending, but the point is that I think what we've learned there is, and again, it's not to just, as Todd said, we don't disparage preclinical models. We have folks that spend a lot of time on those, and in fact, our macrophage targeting agents, the ILT4 and ILT3, came out of those kinds of studies. But we found that, the most effective, it goes back to sort of the basic principles of combination chemotherapy that were worked out decades ago by the, early oncologists. And it's basically independent action, that the best combinations are those when each drug has lots of activity and you put them together, and you generally have good things. So I think, some of the best combinations are ones that we found that you might not have predicted early on, like chemotherapy. There were many that felt combinations with chemotherapy would be immunosuppressive and wouldn't work. And yet those are some of our most effective combinations across multiple tumor types.
Yeah, with the caveat, Eric, that with chemo, things happened in a different way. And it may very well be with immunotherapies when you've got two antibodies working in a similar fashion versus what CTLA-4 antibodies and PD-1 antibodies did, which were a little divergent in the pathways, may be giving us sort of different approaches to what will work in immunotherapy areas.
Could be.
Todd, I don't know if you have anything to add to that.
Not really. No, I agree. Yeah.
I think, and I'll jump in, Alise. I think we also need better markers to profile different aspects of the host immune response. I think we all appreciate you need CD8 T cells in tumors, but a CD8 T cell is not able to do much if you're not able to present antigen, have an antigen, et cetera. So I feel like we need more of a suite of better markers to help us tease out that immune status of the host and of the patient.
Yeah,
And I don't know if any.
Sorry, I will add, sort of pulling on Eric's comment about what we can learn from old combination chemotherapy trials. I think that is really interesting and important. I mean, there's been a lot of recent interest in understanding mechanisms of drug resistance that emerges particularly to targeted therapy, with the idea that if you understand the mechanism of resistance, then you can go chase that and try to inhibit that emergent mechanism of resistance. I think that's not what the old-fashioned combination chemotherapy agents do. As Eric said, when you have fundamentally different mechanisms of action, they typically have fundamentally different mechanisms of resistance. And that, I think, is going to be the key to effective combination therapy, where it becomes mathematically much more difficult for any individual cell to simultaneously develop resistance to two different mechanisms of action. And I think that's going to be more effective than chasing the emergent resistance mechanism, further down the pathway for a given agent.
So Todd, that was a good setup for my next question, because people are using circulating DNA to try and do exactly what you were talking about. Patients are starting to relapse, use your circulating DNA without having to go in and take another tissue biopsy to figure out how their tumor has evolved. And we know that circulating DNA, circulating tumor cells are being used in that way. They're being used to look for early cancer detection with, GRAIL-like assays, and also to serve as an early surrogate for response to therapy or early relapse to therapy. So how good is the technology? Where is it good enough today to be used? Where do you think we need more sensitivity? And how do you see these assays being used in drug development, in the clinic, and even for regulatory approvals? So a lot of questions in there. I think people could take a lot of different aspects of that.
Who wants to go first?
I'm happy to start. We're using, MRD assays are well characterized in multiple myeloma, of course, but we're starting to use that in non-Hodgkin's lymphoma. I think for us, the biggest place it's had an impact is in understanding if patients are going to relapse. What we found is if we have deep, durable, deep remissions early on, that could lead to durability. And if we can monitor patients with an MRD assay and we observe anything at 3 months or 4 months, they're likely to go, the tumor's likely to come back. So we would do a second dose of allogeneic CAR T. But if you don't have any residual disease, you could see durable response. So I think that's an area where we're putting a lot of work into when it comes to biomarkers that can be tremendously helpful.
And do you see, you mentioned multiple myeloma, but there are other hematologic malignancies where doing studies in first-line patients are so enormous and take so long if you use the typical endpoints. Do you see a future where there'll be enough data that regulators are willing to use MRD as an endpoint for approval?
I mean, in my experience, I don't know how much we're seeing change on the FDA front. I think they still rely on a lot of the traditional markers. We hope that at some point they start looking at some of these new markers and make trials easier. But at this point, these are secondary or confirmatory rather than the primary endpoint by which we're getting approval, especially as you're going to think about frontline trials. I think maybe in relapse refractory settings, you could make an argument to use some of these markers at this point. But I think we're hearing encouraging, themes coming out of some of the leadership of the regulators, and we hope that would make trials easier, especially as you're seeing remarkable data for immunotherapy or in earlier lines of treatment. In fact, the lower the tumor burden, the better the data are. So makes an argument for going earlier into frontline therapies, but we need a regulatory framework that supports that.
And Alise, in the solid tumor space, I know many on this panel appreciate how difficult the MRD ctDNA has been. I think one ongoing initiative that's going to generate some very fruitful data is the CT Monitor initiative being led by Friends of Cancer Research. They're in the middle of, we're part of it. I know Merck is part of it, and many companies have come around the table to look at both ctDNA in response to checkpoint inhibitors as well as the TKIs. And I think it's a brilliant, well-conceived project. All the companies are coming together, transparently sharing data. And a lot is going to come out about, I think, also about early response and how that translates into later effects for patients and benefits.
So Alise, you also asked about are the tests technically where they need to be? And I think the answer to that is going to depend on the particular type of application. So if you think about it in the, screening a healthy normal population for occult cancers, I'm less worried about the technical performance, the sensitivity of a test. I'm more worried about the public health implications of screening huge populations and what happens in follow-up and the morbidity that could be associated with that. And I think it's almost certain that healthy individuals are going to have small cancers that are not life-threatening. And I'm not yet totally convinced that detecting them is going to be a good idea. Indeed, a lot of the cancer screening that we have to date suggests that it may not be a good idea. Then on the MRD side, I think that's going to be really important in a number of different ways that we've just been discussing over the past few minutes. And there, I don't think that the currently available tests have the sensitivity and accuracy you'd really want to have. And I think that it is becoming possible to make those even better, 100-fold more sensitive to detect MRD. And I think that's going to really have a profound effect. Getting back to the resistance question, while it hasn't been shown yet, I don't believe that earlier intervention is beneficial to patients. If you just think again, if you can reduce by orders of magnitude the number of cells at risk to develop resistance mutations, the probability of a resistant clone is going to be smaller. So I'm pretty confident that it used in the right way with the best tests, and of course you need effective drugs, that bringing MRD into the clinic is going to have a huge effect. Whether it's an approvable endpoint, I don't know, but as a way to guide, which clinical trial strategies to double down on versus which ones don't look promising, even if the FDA doesn't look at it at all, I could see drug developers really using this as a really powerful set of tools.
Eric, I don't know if you want. Yeah.
I'm just saying, I think Todd's spot on there. I'm not sure that the regulators are going to agree, but it's going to be a way to drive proper clinical development. Remember, tumors are the immune system's jobs to tell the difference between what's you and what isn't you. And tumors look a lot more like you. So that's why, getting rid of the dampening effects of tolerance with anti-PD-1 antibodies, et cetera, has this dramatic effect that we've begun to see over these last years. And so our job is to figure out how to use MRD and a lot of other tests to be able to play the right games with different tumors in different ways so that we can make the immune response, see the tumor as foreign enough to get rid of it. And the earlier we do it, the better, because when you have fewer cells for the immune system, for NK cells, for innate immunity, for T cells to see, the easier it is to clear things up and not worry about MRD.
Yeah, so Lisa, I think the other panelists have really covered it well. I don't really have too much to add, other than again, this is a very important area that we're involved in all the sort of, I guess, use cases that you've mentioned, and as Christine said, I think this Friends of Cancer Research effort, I think is a nice example of industry and other stakeholders, academics, FDA coming together, trying to gain evidence that would enable use of ctDNA as for a regulatory endpoint.
I'll point out I'll highlight one example where I don't know if circulating tumor DNA was used in decision making, but it certainly may have been. Very often in phase 1, we say stable disease. We don't know what to do with that in a non-randomized study. We're really looking for responses. And yet there was a recent drug for uveal melanoma. Really, it was mostly stable disease, very few responses. And I thought they were quite bold. They went into a randomized phase 3 study, which was spectacularly positive with a significant increase in survival. And really, if you go back, and then they went back and looked at the circulating tumor DNA, there was a profound decrease in that, and that predicted survival. So I don't know if that was used in their decision making, but it certainly may have been, and I think led them to what was, not throw out a drug that actually turned out to be quite efficacious. So I think it could really expand the number of drugs, hopefully, that we take into successful late line late line studies. We've only got a few more minutes left. One, I don't want to forget about access. And I think Andy talked about this a little bit. Access in clinical trials, diversity in clinical trials. Andy talked about this in his opening remarks. And maybe those of you who are involved in clinical trials might comment on what your companies are doing to try and increase diversity in oncology clinical trials.
So I'm going to speak briefly about that from the point of view that while it is incumbent upon companies to try to get diversity in the clinical trials, it's also the way we choose our investigators because of the investigators' access and ability to create a diverse population within their own piece of what we're doing. It is not easy. And it's something that we have to do. One of the things that's clear, and we see this in society, is not only are clinical trials not diverse enough, but when we get drugs out there, the ability of access, as Andy said earlier, isn't what we want it to be. And it's that's true within the Western world. It's also true when drugs don't get to the rest of the world. And this is something that we've got to do moving forward, but it's going to be harder work than anyone can imagine.
So Alise, I'll chime in. I mean, I think I know we're short on time. It would take me quite a bit of time to go through all of our efforts. This is a very important topic for us. But one of the things is that I think this is a system issue, and I think, like probably many on the panel, we're working with various individuals representing different constituencies to try to solve this. One of the things that to me was interesting, it was a meeting at ASCO where there was a speaker from an academic institution who mentioned that some of the issues are getting information to folks, right? A lot of people don't have a computer. They're not able to access Google, right? But the interesting thing is pretty much everybody has a cell phone. Even in rural communities, everybody has a cell phone. So they're working on enabling cell phone-based information to get out to patients related to clinical trials, for example. So I think, there has been progress here, but it remains an important area.
Thank you. There have been a bunch of questions online, and I know we're up at our time limit that talked about biomarkers, and I think questioned whether drug companies were really committed to developing drugs in oncology and biomarker selected patient populations. I know from my experience, we definitely have been committed to that, in that it's the best way to get a successful drug is to go after the patients who are most likely to respond to it. So I know from my perspective, if there is the view out there that drug companies are no longer doing it because they want to go into large populations, I think it's really now we're at the point where it's just a reflection of how hard it's been. And I don't know if anybody else wants to comment on that on the panel.
When you see the mechanisms that we're targeting, right? It's, the multiple KRAS inhibitors, there's just so many different combinations now. I think we have no choice. We have to do this, otherwise we won't be successful. And I think in the end, it's the patient community that benefits.
Yeah, the other thing, Alise, I'll just say is that, again, it may be a small population in one tumor type, but we just had yet another tissue agnostic approval yesterday for a combination BRAF MEK inhibitor. And I think when you start, when across every tumor type, the numbers are not so small anymore. Same was, of course, with the one we worked on for MSI high.
Okay. Thank you, everybody, for participation in this great panel. Apologize for the technical difficulties in the beginning of it, and I think we'll turn it back over to the next panel.
Thank you, Alise.
Thank you all. Thank you, Alise.
Thank you, Alise.
Thank you.
Perfect. Thank you very much to the panelists and to you, Alise. You're demonstrating our third year into virtual working, the need to have at our fingertips compensatory processes and mechanisms when our our first doesn't work. So terrific panel. One comment, actually, you mentioned Immunil, a company that's been working to bring affordable autologous CAR T therapies into India. And actually one of the founders of Immunil, Kush Parmar, will be with us at the India clinical trial panel. So we really encourage you to participate in that.